Hello guys, and welcome to this video on cellular adaptations. I'm going to try to explain this topic in the most concise and simple way. A really important basic principle is that our organs are under constant stress and they're in balance with that stress. And when there's a change in the intensity or type of stress, cells can adapt. This adaptation can be normal or abnormal depending on the stimulus. And even though cells can adapt, if the stress persists for too long or is just too intense, this can lead to cell injury and eventually cell death. There are four main cellular adaptations. The first one is hypertrophy, which is an increase in cell size. The second one is hyperplasia, which is an increase in cell number. The third one is atrophy, which is a decrease in cell size and number. And the fourth one is metaplasia, which is a change in the type of cell. Hypertrophy is an increase in the size of a cell. The mechanism by which it occurs is that there's a stressor, and this stressor activates certain genes, and when these genes are activated, they increase the production of cellular organelles and proteins. Usually hypertrophy occurs together with hyperplasia because they have similar stimuli. And some examples of physiologic hypertrophy include the uterus during pregnancy and muscle growth when people go to the gym. A pathological example of hypertrophy is hypertrophic cardiomyopathy. This is a disease that usually occurs due to chronic hypertension. I think this is a good time to talk about the types of cells in regards to cell division. We have three main types of cells. Permanent cells, from their name, never divide. They're always in the G0 phase of the cell cycle and never leave it. They include neurons, skeletal muscle, and cardiac muscle. This is important because permanent cells only undergo hypertrophy in response to increase in stress, because they can't undergo hyperplasia. The second type of cell is called stable or quiescent. These cells normally are not dividing. However, if they are stimulated to divide, they can divide. They include hepatocytes, lymphocytes, periosteal cells, and many more. The third type of cell is called labile. These cells are always dividing. They never enter the G0 phase of the cell cycle. They include skin, hair follicles, the epithelium of the gut, bone marrow cells, and germ cells. An important tie-in to this point is that chemotherapeutic agents tend to affect labile cells the most. And this occurs because the mechanism of action of chemotherapeutic agents usually involves something in the cell cycle or something that happens during cell division. And because labile cells are always undergoing cell division, they're the most affected. That's why many chemotherapeutic agents cause hair loss. That's why many chemotherapeutic agents cause diarrhea, bone marrow suppression. Okay, moving on to hyperplasia. Hyperplasia, by definition, is a controlled process. It involves creating new cells from stem cells. Usually occurs due to hormonal stimulation, and some important principle in pathology is that any cell that is constantly dividing has a higher chance of turning into cancer. And pathologic hyperplasia can progress to something called dysplasia, which is disordered cell growth, and eventually can progress to cancer. Some physiological examples of hyperplasia are bone marrow hyperplasia in response to anemia or decreased blood. Let's say someone donates blood. In response to that donation, the bone marrow is going to increase the production of red blood cells. This is the normal response. The uterus also undergoes hyperplasia during the menstrual cycle. Some pathological examples are endometrial hyperplasia, which occurs due to chronic estrogen exposure, and prostatic hyperplasia, which occurs due to chronic androgen exposure. 2 slides ago, I stated that hyperplasia usually causes an increased risk of cancer. A very important exception to this rule is benign prostatic hyperplasia. Benign prostatic hyperplasia does not increase the risk of prostate cancer. Atrophy is a decrease in tissue mass. You can reduce the tissue mass by reducing the number of cells or reducing the size of cells, and they usually occur together. Many things can cause atrophy, including decreased blood supply to an organ. When a nerve is transected, the muscle that it's supplying becomes atrophied because there's less stimulation. Decreased hormonal stimulation can cause atrophy of that organ, and poor overall nutrition can cause atrophy. It's very important to understand the mechanism by which atrophy occurs. The number of cells gets reduced, and this happens via a process called apoptosis, which is programmed cell death, and the cells that are present have a smaller size. To reduce the size of a cell, there are three things that are happening. First of all, the cell synthesizes less proteins. The second thing is that the cell breaks down the proteins that are already present, and this occurs via a very important process called the ubiquitin proteasome pathway. To understand this, we need to first understand what ubiquitin is and what a proteasome is. Ubiquitin is a small protein molecule, and proteasomes are complexes that have a function of breaking down proteins. So what happens in this pathway is that ubiquitin gets added to certain proteins that the cell wants to get rid of, and then these proteins that have ubiquitin tagged with them get recognized by proteasome complexes, and proteasomes take them down and they break the proteins that are tagged. The last thing that happens is that cellular organelles get digested by the cell. This process is called autophagy, and what happens is that the cell starts to form vesicles around the organelles that it wants to destroy, and then these vesicles fuse with the lysosomes, and the process of a vesicle fusing with a lysosome forms something called an autophagosome. The lysosome then releases its hydrolytic enzymes, and the enzymes break down the organelles. Moving on to metaplasia, which is a change in the cell type. If you want to change the type of cell, you need to go back to its stem cells and reprogram them. So the process of metaplasia involves reprogramming of stem cells. Usually, what causes metaplasia is a chronic irritant, and the whole point of changing the type of cell is that these metaplastic cells are supposed to be better at handling the irritant. However, the problem with this is that metaplastic cells have a much higher chance of becoming dysplastic and eventually turning into cancer. A classic example of metaplasia is something called Barrett's esophagus. Normally, the esophagus is lined by non-keratinizing squamous epithelium. People that have gastroesophageal reflux disease have chronic reflux of gastric acid into the lower esophagus. This chronic irritation of the lower esophagus by gastric acid causes reprogramming of those stem cells to produce a type of epithelium that can better handle that acid. So what happens in Barrett's esophagus is the squamous epithelium gets turned into non-ciliated columnar epithelium with goblet cells. And these cells can produce mucus, which can neutralize the acid. However, the problem with this is that Barrett's esophagus increases the risk of esophageal adenocarcinoma. Smoking is another very important cause of metaplasia. Normally, the bronchi in our lungs are lined by something called the respiratory epithelium, which is ciliated columnar epithelium. And this specific type of lining has a very important function in clearing waste via something called the mucociliary escalator. In people that smoke, chronic irritation from the smoke causes metaplasia into stratified squamous epithelium, which is generally more durable. However, when this happens, the mucociliary escalator function gets lost, and there is also an increased risk of developing squamous cell carcinoma. Metaplasia can also occur due to vitamin A deficiency. There are areas in our body that have very specialized epithelium, such as the conjunctiva. And vitamin A has a function in maintaining these specialized epithelium. So a deficiency in vitamin A can cause metaplasia in those areas. Metaplasia can also occur in connective tissue, and an example of this is myositis ossificans. What happens here is that skeletal muscle gets turned into bone. This usually occurs after trauma, such as surgery, and as the muscle heals, it gets turned into bone. Because I said that hyperplasia and metaplasia can eventually lead to dysplasia and cancer, I want to talk a little bit about dysplasia. Dysplasia is defined as disordered precancerous epithelial growth. And for you to call something dysplastic, it needs to be epithelium, by definition. This isn't considered an adaptive response. And because dysplasia is precancerous, it's very important for you to be able to recognize dysplasia under a microscope. There are two main changes that happen in dysplasia. First of all, pleomorphism. Normally, epithelial cells that are beside each other have a very similar size and a very similar shape. In dysplasia, cells that are beside each other lose this uniformity, and this is called pleomorphism. The second thing that's important to recognize is that nuclear changes occur. Chromatin becomes clumped, and the nucleus to cytoplasm ratio increases. Dysplasia isn't considered cancer because dysplasia can be reversible. However, if dysplasia involves the whole thickness of the epithelium, it's generally non-reversible, and we call it carcinoma in situ. This concludes the video on cellular adaptations. Thank you for watching.